our biomarker project. This is actually largely work I'm going to sort of largely credit Dr. Anna Falzer here, who's my mentor at Combined Brain. I'm a postdoctoral fellow at Combined Brain. But Anna is really has been leading this biomarker project. She's the head of our biorepository. Any blood collections that, if any of you were here 2 years ago in 2023 when we did those for Dirkwinney, Anna was the one really leading that charge. She was not able to make it today because she's at a different conference for Cool. I think she's at Coulin-de Vries syndrome today. I've lost track. All right, so I'll just do that. All right, kind of a brief overview of Combined Brain. We all like to show this slide a lot. But so Combined Brain, we're a consortium of patient advocacy groups. We kind of work as scientific advisors and coordinators, and we want to help each disorder, Dirkwinney in this case, really get everywhere from the very beginning stages of identifying the causative gene associated with the disorder, all the way through getting a drug development or gene therapy development, clinical trials. And kind of everything that you see above this line here is very patient-focused that we work with. A lot of what Oliver was just talking about, where we try to help people get natural history data. We do disease concept studies and look at really patient-centered outcomes. So what outcomes really matter to patients? What can we measure improvement in or lack of improvement in that really actually matters to the families? And then everything below the line here is more basic science focused. And today I'm going to talk about biomarkers in particular and kind of the biomarker wing that of Combined Brain that we've been working with. So what is a biomarker? A biomarker is an objective measure which can be used to predict disease status, progression, and or response to treatment. These can be mRNAs, epigenetic markers, proteins, metabolites, even microbiota. And these are just coming from biological samples that can be found in any really biofluid, from cerebrospinal fluid, blood, urine, saliva, stool. And really the advantage of biomarkers that are coming from these biofluids are that these are often non-invasive or relatively non-invasive ways to try to measure outcomes in patients. So a good example of biomarkers that's been identified is NFL, which is neurofilament light, that has been identified as a biomarker of Huntington's disease, which Anna has extensive experience with. And so this graph on the left here, you can see that as NFL concentration in blood plasma increases on the y-axis, it corresponds with blood NFL concentration in CSF. So this kind of confirms that the blood plasma in your cerebrospinal fluid can be measured accurately just with a simple blood draw. And that as baseline plasma NFL increases, this correlates with white matter atrophy in Huntington's disease. However, we have a very limited access to patient samples with rare disorders because a lot of these patients are very severely affected. It's very difficult to travel. And so there's really just a scarcity of good biosamples from patients with disorders such as Dirkwinney syndrome. So here at Combined Brain, okay, I'm going to actually skip to this slide and then skip back. At Combined Brain, we're working to try to fix that. So we have a biorepository. It's held at Van Andel Institute up in Michigan. And one of the main things we do every summer is that we actually fly out to family conferences such as this one. We flew out to the DYRK1A conference 2 years ago in 2023. And we fly our research team, we fly out pediatric phlebotomists, we fly out all the different supplies needed for blood collections, and we come to you. We can also do home visits where we actually have contracts with mobile phlebotomists. A nurse will come to your home and coordinate blood collection visits. And we collect everything, blood, stool, even brain samples from post-surgery, all of the above. So we had this study design where we wanted to look for biomarkers in these rare disease patients. And so we had this aim of aiming for about 20 affected individuals with about 20 to 40 to actually neurotypical controls. These are usually familial controls. We tried to aim for age and sex match controls. So siblings are our primary control, but parents if there weren't enough siblings available. We collected blood plasma from both of these populations. We also had these patients fill out surveys, namely the Vineland-3. This is a measure of adaptive behavior, as well as the PELS, the Pediatric Epilepsy Survey. And we processed the plasma for proteomics, looking at all the proteins in the blood plasma using SomaLogic, which is a company that does that. And we looked broadly across the board. And then we compared both proteins, differences in expressed proteins by disorder, by control versus affected. We looked at proteins compared to age, and then also proteins by epilepsy, non-epilepsy, and we're beginning to do some network analyses. So for Dirkwinney syndrome, from the samples that we ran this proteomic screen on, we had a sample size of 15 affected patients with an average age of 6.5 years old. The percent female was 46.7%, telling you that we had a really good ratio, about 50-50 of males to females. 73% of the people we collected plasma from reported that they had epilepsy. And this is the adaptive behavior composite from the Vineland. So the scores here, 50, 51, and 49, this is communication composite and daily living score composite. So for the Vineland, about a score of 70 is considered kind of normal. So it looks like Dirkwinney kind of scored within a very comparative range to many of our other disorders that we collected from for this study. So here is that Vineland-3, the Vineland-3 scores. So if you look at the adaptive behavior composite, again, this blue line here is right at the 70, a score of 70. And so we had this is across all disorders, we had a really broad range of a broad range of scores. But if you compare with the chronological age here, so, ages 1 to 20, but also the age equivalent, if this is our age equivalent score, all of the disorders which we analyzed had a lower score where you have this slower increase in gain of skills over time. And then if you pull out Dirkwinney specifically, here's Dirkwinney. Again, this is limited. I should have pointed this out earlier. This is limited by, we actually condensed down from up until age 18. We had some Dirkwinney patients who were above 18, but for the sake of simplicity, we noticed that our above 18-year-olds were kind of outliers. So this is without those outliers. Similar for communication composite. This was a little bit more even across the board in terms of all of our different disorders. And then again, Dirk1A, you kind of see this plateau, at least in our Vineland scores as we collected them. But as Oliver said, we always want to collect more Vineland scores via RareX so that we can really make sure that this is truly representative. If you look across disorders, there's really no distinct changes in plasma proteomics by sex, age, or epilepsy burden. So you're looking at a U map here. So this is really just a measure of how different are these two factors compared against each other. So if you compare males and females, what you need to see is that blue and red is really just scattered throughout. Each of these dots stands for an individual. Same with age. There was really no clear segmentation by age or by epilepsy, yes or no, if you just look at all versus all. Also, if you go by disorder, here are all the different disorders which we did our proteomics on. And again, you don't see a clear clustering. So Dirkwinney syndrome here is kind of this light blue dot. It's very tiny. I'm sorry to those in the back. But it doesn't seem to cluster really with any of the others in particular. If you take a look at all the different blood proteins which were enriched, you can see that here in red is everything that was enriched. And this is positively and negatively enriched in our affected patients versus blue is our control population, which was enriched. So some common proteins of interest by disorder started to pop up. And I'll show you a graph of this in a second, but a few that I want to focus on are these SAA1 and 2, GFAP, which Oliver mentioned, and HBG1. These two being significantly downregulated in Dirkwinney syndrome. GFAP, this doesn't look significantly up right now, but that's because this particular graph is showing you without a couple of the outliers, or not outliers, a couple of the age outliers, which we need to build out towards. This is only up to age 18. So SAA1 and SAA2 is serum amyloid A1 and A2. These are highly expressed in response to inflammation and tissue injury. And we've had high levels being found in inflammatory diseases such as arthritis, Alzheimer's disease, other diseases like that. Glial fibrillary acidic protein is a very common biomarker used for neuronal stress and damage. You see it in Alzheimer's disease as well as Parkinson's disease. And then HBG1 we were kind of surprised by, but it's found to be a biomarker specifically for blood disorders such as sickle cell disease or beta thalassemia. We don't really know what this is indicating yet. This is actually a fairly strong hit across many of our different disorders. So other than our initial findings of GFAP, which we found at our initial roundtable, we've actually found more and stronger biomarkers since our last update. So if you look at just differentially expressed proteins in Dirkwinney syndrome alone, this is with those tighter parameters. This is a volcano plot showing that in red here are positively enriched blood plasma proteins, where you see HBG1 here. And then we have some negatively enriched proteins as well. And here's just the graph with everything without those tightened parameters. Here is GFAP with, look at how greatly increased these are with HGB and SAA1, SAA2 here. So those are our initial findings. Anna and the rest of our team are working hard to write this up as a sort of cross-disorder publication, which we're hoping to submit very soon here. But our further research is that we are going to do a round 2 where we take some of these proteomic hits and we're going to actually do a second round of more targeted proteomics for especially a smaller set of patients, a smaller set of proteins, and really confirm these results. And hopefully with more blood collections in the future, and as we continue to collect more survey data, we can draw stronger correlations to make this a true biomarkers of Dirkwinney syndrome. And again, the kind of advantages of biomarker is that then, therefore, going forward in clinical trials, eventually, this can be kind of used as a readout for was there improvement or not in this patient, given this therapy. So a big thanks to all of our Combined Brain biorepository team. It's a lot of work, a lot of hands-on, just collecting blood samples. Parts of many of our team like to dress up as Mario characters just to kind of help the kids because it's very scary to get a blood draw at a conference. You're in a new place. So it's a lot of hands-on team coordination and effort. Thank you to our different funding support. We worked together. This is a pre-competitive plasma proteomics project, so we had industry funding to really jumpstart this. And then hopefully these industries, they chose the disorders that they were interested in and wanted to see plasma proteomics for. So it's kind of a win-win situation for everybody. Thank you to all of our different disorders that we worked with. And I just have to throw this out here. For families, please, here's my obligatory plug. We have two, RareX natural history studies, but also 2 new surveys that I would love to encourage people to fill out. I know it's yet another survey. I have flyers for each of these at the table out there, but gastrointestinal issues are a huge issue for people with Dirkwinney, and we're creating a survey to objectively measure that. And then this patient priority survey was developed in tandem with our disease concept model. And this survey is to basically validate all the findings in our disease concept model so that we can then publish our disease concept model. So this will really round out that study that's been going on for over a year now with Olivia Salverson, a master's student in genetic counseling. So thank you, everybody.
I could raise my voice. That was really nice for question. So as I'm sure you know, APG1 basically points to a field hemoglobin.
Mhm.
So do we know about red cell parameters, altered syndrome, and
I think that might have to be a next step. I do not know of anyone who has specifically looked at that specifically for DYRK1A. But yeah, thank you.
Very straightforward question. I know your sample size is small, but I use like GCD nation severely biomarker.
We have not done a genotype specific analysis. We've been working on sort of the genotype phenotype correlation. It's difficult without doing, I mean, if you really want to go all in, you could do a complete functional screen of every single DYRK1A mutation against, correlating with the phenotypes that we're seeing in the Vineland. It's very, very difficult to predict severity of the functional consequences, severity of that compared to genotype just by seeing the genotype. It's not as clear cut for DYRK1A. I've done some analysis, just kind of, lining up all of the different variants across the gene and trying to see if we can correlate. But there's no clear answers right now. But that's the ultimate goal, yes.
Thank you for the very interesting. Can you just detect tau in the realizers? Because tau is a target of checkpoint inhibition, and that is stated that this is not that in the education after that is
Yeah, I took a look at that, I believe, months ago now, and didn't see any strong difference.
Did you look at the phosphorylation?
No. Mm-hmm. Yeah, really quick, our aptamers, I think, are not specific enough in this large somologic screen to really tease that out, which is why we're doing a round 2 that's more targeted. I'd have to ask Anna, but that might be on the short list for something to look for. Yeah, sorry. Hello.
Wondering when you compare between disorders in these biomarkers, do you expect to find biomarkers that are specific to each gene, or do you think that there may be some that can be biomarkers for multiple of these disorders, and then is that being reflected in the biomarkers?
Yeah, I think it's probably realistically, I mean, in a perfect world, we would have a specific biomarker per disorder. But in reality, I think we're really going to, which is what we're finding. We're finding a lot of these kind of broad inflammation or just general, disease-associated markers that may not be specific per disorder, but still, as long as we can get a strong enough read of that biomarker per disorder, GFAP or NFL, which we spent a lot of time looking at as well, but didn't see a strong, kind of surprised by. Yeah, as long as it can be used as a readout for some sort of clinical study, that's what matters more than it being specific to the disorder.
Yeah, I can have a follow-up question to that. So you mentioned that HBG was in DYRK1A and it was also in other disorders. Like, how many other disorders was it involved in? Because I think that part to me is kind of interesting. That it seems like this is, and subtly in gamma. I mean, like, why would that be all these different disorders?
Great question. I think because again, Anna would have a better answer perhaps than I. We actually just found this, I think, only a couple of weeks ago. So this is kind of hot off the presses. We haven't fully thought through this. So please tell us what we can investigate with this, because we were all surprised. I had just for this side, for simplicity's sake, for this slide, I had just highlighted, DYRK1A, but you can kind of see which ones look more obviously dysregulated. DYRK1A was unique where it was down. It looked kind of down, I don't know, versus others which were severely increased. Yeah, I'm not sure. I'm just not sure.
And then I think you showed one figure. It was like, I think on my chronological figure, And it looked like one of the disorders still looked like it was like fairly linear. Is that like SYNGAP? Because it looked like there was the other not in.
This one, yeah, that looks like SYNGAP, yes.
So that's what, so it seems like it's somewhat different than the other disorders of age, and like if you remove that, process, like would that change it at all? Looks like that one's like a little bit less like the other.
Less severe. True, but if you look at expressive communication by age, you see that CTNNB1, and I think that's maybe CACNO, I can't tell the shade of pink on this slide. CACNO1A right next to it, also you see, it's lower, but it's the same slope of the line essentially. Yeah, I mean, for DYRK1A, everything that I showed was DYRK1A specific. I would actually be really interested in seeing what their specific their volcano plots look like. I've glanced at them, but not really looked at them.
I won't try to say this the best I can, but you pointed out one of the biomarkers of being higher in year one, you talking like in age to year one.
Yes, yes.
Would that correlate with the development of higher biomarkers?
Yeah, so
When it developed.
yeah, so what we're showing, a lot of these, what I was showing today is adjusted for age. That's why we try to get age and sex matched controls to compare directly against. And a lot of our earlier analyses, we were just kind of grouping together all the ages, but then as we've consulted with, thought about this for months, we've started really adding an extra, I guess, controls for age. So GFAP seems to kind of jump with age a little bit, I believe. And so our earliest results, GFAP looked really hugely like a strong biomarker, and now it still is, but it's a little bit weaker because we're adding in age as a factor in our statistical analysis. Is that answering your question? Okay.
Sorry. Are you taking notes on medication status for these ones? And is anything coming out that's related to medication status rather than?
Yeah, we do ask for, when we collect blood, we ask for medication. We even ask for, are you on a special diet? Even when did you last eat? But I don't know if we've done that analysis yet, to be honest.
I think on our schedule, we have a break right now.